I don't know whether there are any Greeks in the audience, but I'm told that our job as physicians is to remove arrows. But unfortunately, many of the life-saving interventions in critical care we have learned over the past decade or two actually point arrows at the lungs. And you've heard a fair bit already about the topic of this symposium, that is ventilator-related lung injury. One of the messages that I also would like to leave you, which I think is very important, is that we ought to be always very specific when we talk about the term injury. Injury means different things to different people. And there are various manifestations of injury. As clinicians, we integrate the information, but as we get more into reductionist biology, our endpoints become more specific. And these different manifestations of injury are loosely correlated with each other, but they are only loosely correlated with respect to space and time. The genomic response does not occur necessarily at the same time as edema formation or changes in mechanics and so forth. And therefore, all of these are manifestations of injury, but there are specific manifestations of injury. So when I'm talking about injury, my focus is going to be on individual cells, in this case lung cells. We've known for a very long time, starting with the defining studies by Didier Dreyfus, subsequent work by John West's group, and more recently John Marini, that the cells of the alveolar capillary barrier of the lung have characteristic morphologic changes in certain models of ventilator-induced lung injury. And the model that I also will talk about and show you evidence from is the classic hyperinflation overdistension model, which on the surface, as Dr. Slutsky already mentioned, may sound very unphysiologic when you go ahead and mechanically ventilate a normal lung with pressures and volumes that a rat would never experience during life. But if we think about the kind of redistribution of mechanics, the de-recruitment, the flooding and the collapse of lung units in injured lungs, certain parts of the lungs may well experience the equivalent of a vital capacity maneuver on every breath, and that's after all what's being modeled in these models. So I only want to point out that some of the changes that we're talking about is there appear to be gap formations both between and within lining cells, here alveolar epithelial cells, but the same could be shown for endothelial cells of the thin part of the blood-gas barrier. There is bleb formation where the plasma membrane lifts off the subcortical cytoskeleton. And these, as I will show you, are manifestations not just of injury, they're manifestations not just of cell necrosis, but they're also manifestations of cell adaptation and repair. I greatly benefit from the previous presentation in as such you all know that the lung is a network structure that's internally distorted by surface tension. So for a given lung volume, there is a rearrangement of the dimensions of the stress-bearing elements between alveolar duct and alveoli depending on the surface tension. So this network structure is distorted by surface tension. The other thing that is useful to remind ourselves about that this is an exceedingly vascular structure. So the stresses that are applied on the lung, it's not too surprising that they're associated with the accumulation or the egress of fluid from the capillary into the alveolar space, which then in turn inactivates or alters surfactant function, causes all kinds of barrier properties, mechanical consequences, immune responses, and so forth. For the purpose of my talk here, we ought to look at this structure and appreciate that these stress-bearing elements are decorated with cells. The cells themselves don't need to carry much stress. It's very easy to push and pull on a cell. But nevertheless, the cells still need to cover and move along the structures during each breath. And if the area that an alveolar epithelial cell, for example, has to cover increases a lot on every breath. And that is why the discussion that we just had, how an alveolus changes size during a breath, is very important. So if there is a large area change, that imposes a large shape change on the cell. And so the question then is, well, what does a cell do when it's exposed or when it's forced to assume a new shape? In very general terms, cells are spot welded to their matrix. This is a cartoon of a plasma membrane, adhesion receptors, a focal adhesion complex, which are up to 50 proteins, which link the adhesion receptors, integrins for example, with tension or stress-bearing elements of the cytoskeleton, the actin cytoskeleton for one. That in turn is being tensed by molecular motors. And so when there is a stress or an area change or a strain, a deformation, and incidentally, we're sometimes forgetting that one really cannot uncouple pressure from deformation, force from strain. These are interlinked quantities. So to some extent, we ought to be careful when we're talking about whether there is a pressure sensor as opposed to a strain sensor. The way a cell senses anything requires some conformational change of some molecule in the cell. There is no, without that, there is no signaling. And so in part, when we're talking about pressure versus strain, we also need to talk about at what scale we're looking at this problem. But in general, you can think about this focal adhesion complex, this family of 50 proteins, like a little strain gauge. So when the tension here increases, or when the matrix, the basement membrane, gets deformed, proteins in this strain gauge alter their shape. And that opens up pockets for interactions with signaling molecules, for changes in activity state. So actually, I don't need to even think about stressing the plasma membrane, and I could get a deformation response, which fundamentally is related to this strain gauge. One, of course, doesn't exclude the other. If, for example, I have a very round cell, and I change the basement membrane area to force it into a pancake, there comes a point where there will be tension in the lipid bilayer of the plasma membrane. That tension in the lipid bilayer could well become lytic, and then the bilayer busts. Or before it becomes lytic, it may open an ion channel. Calcium goes in, triggers all kinds of signaling, and ultimately a genomic and proteomic response. So as Dr. Uhlig has already told us very elegantly, there are a variety of parallel paths that can lead to signaling in the cell in response to deforming stress. Incidentally, this insert here is a A549 cell. It's an alveolar epithelial cell. It's really a cancer cell. It's a cell line. But the point of it is one of these proteins, paxillin, was coupled to a fluorescent marker, so that you can see that this cell is not simply glued to the matrix, but that the cell has individual spot welds. And these spot welds you can actually show grow or shrink based on the force the cell or the surrounding puts on these spot welds. So when we're talking about cell mechanics, in contrast to some inert material, a piece of steel, what dominates cell mechanics is not just the inherent mechanical properties of its cytoskeleton and the covering plasma membrane, but also the rate and extent to which cells can remodel their stress-bearing elements over the time you apply the stress. So if you stand there and I push you, you would fall over if you didn't have a corrective action, step backwards and so forth. The cell remodels on a temporal scales of microseconds when you deform it. And so that becomes an important determinant of the likelihood of stress failure of the system. Now, the signaling or the potential signaling pathway that I'm going to focus on, or the events that I'm trying to focus on, have to do with cellular stress failure. And so a number of years ago, Ogi Gaich in the lab, and I realized that the slides are getting somewhat compressed here, set up an isolated perfused rat lung model to ask the questions, are these cytopathologic changes that we've learned about from electron microscopy evidence of plasma or cellular stress failure in repair or not? And what he did is, while the lung was mechanically ventilated with either injurious or non-injurious settings, he added a membrane impermeant label to the perfusate. And that membrane impermeant label, propridium iodide, when it meets either RNA or DNA, can be excited to fluoresce red. So every red dot is the nucleus of a cell with propridium iodide. And the fact that propridium iodide gained access to that nucleus must mean that the plasma membrane, over the course of this experiment, at any one time must have broken. Otherwise, the marker wouldn't have gained access to the lung. And so you can see that there is the light microscopic correlate of injury, and that when you mechanically ventilate this preparation with arguably quite injurious settings, then this is more a demonstration project than asking a question whether this setting is injurious. You can see there is quite a remarkable amount of cell injury here. What you see in blue here is autofluorescence. You see the structure that Dr. Niemann has already pointed out to us. This is an optical slice of an alveolus obtained with confocal microscopy or of subpleural membrane, about 20 microns below the pleural membrane. And not surprisingly, as you go from admittedly minimal or non-injurious settings to more injurious setting, this marker of injury increases. Well, what happens to a cell when the plasma membrane breaks? Well, one of two things. Either calcium goes in, and unless the cell repairs the wound, the cell will die. And probably it has about a minute or two to accomplish this task. If it doesn't do that, the cell will undergo death by necrosis. But as I will show you, most of the time, even in as harsh an experimental setting as the one that I've just described, there is wound healing by one of three mechanisms. The energy of the free edge of this wound drives lipid molecules towards the wound, so to close the wound, bilateral lipid flow. But more often and more effective is the fusion of vesicles that sit under the plasma membrane. So the cell and organelles of the cell supply lipid to fuse the hole, or patch formation, which move by site-directed exocytosis to plug the hole. And as and after that has happened, there is a genomic response so that stress genes are turned on, leads to pro-inflammatory signaling, and it is a priori not clear whether we should be happy that the cell healed or the cell died, because this cell could turn into quite an angry cell, which then sends signals to its uninjured neighbors to perpetuate and amplify the pro-inflammatory response. So this can become quite an important signal transduction event in ventilator-injured lungs. That cells heal has been appreciated for some time, and this is a very large cell. This is a sea urchin egg. And it's large, so you can see what happens. So when you cap the top of a sea urchin egg, there is this lipid trafficking response. The hole is plugged. And the point of this example is that this is not a lethal event. This egg divides happily. Function is preserved, even though a rather large wound had been imposed. And if we look at mammalian cells, virtually all mammalian cells, to varying degrees, have retained this important ability to repair their defects. Is there any clinical correlate to this? You can go ahead and measure troponin levels in patients with sepsis, and you find that they're elevated. Well, that doesn't mean that these patients have myocardial infarctions. What it tells you is that a protein that is usually confined to the myocyte has leaked into the circulation. And it's evidence of plasma membrane stress failure mechanically in the heart. And in this rabbit model of aortic banding and stimulation of the heart with isoproterenol, you can show that 40+% of the myocytes in the heart took up this marker that was coupled to albumin. And in fact, the function of the heart at this point in time, even though this event has happened, was perfectly preserved. But you could imagine that there's going to be an inflammatory event and secondary consequences to this. We could have examples of this in the GI tract in response to what we eat all the time. I mean, your gastric cells are injured mechanically all the time. So this is a rather important healing and response. So now let's ask ourselves, well, do lung cells, whatever they may be, endothelial cell, epithelial cells that are injured during high-volume ventilation, do they heal? And in order to answer this question, we compared the amount of cell injury as the number of red dots per alveolus in an experiment when the lung was labeled during injurious ventilation relative to the amount of injury that was apparent when the label was only applied once we were done and the lung had been returned to non-injurious settings. So the duration and amount of injury is the same in both instances. But here, if every cell here had been necrotic, we still should have been able to label the cell at this point in time. And as you can see, there is much less injury or apparent injury when you try to label the lung after remove the injurious stress. And that is consistent with work by West's group, for instance, showing that the sieve-like injury or the filtration coefficient of the lung, which increases in response to mechanical stress, normalizes rather rapidly after one removes the mechanical stress. And the same thing can be shown in frog mesenteric vessels. This is a general feature of vascular and cell repair. The kind of organelles that are recruited to the cell surface during this repair response are manifold, but the ones that have received most attentions are lysosomes. And it's been argued that this is a primitive host defense response against invading organism. As you may recall, lysosomal content is very acidic. And it's been postulated that the fact that lysosomes contribute to the patch formation is helpful in the defense against invading microbes. So, how do cells maintain sublytic plasma membrane tensions? What do they do to prevent this from happening in the first place? And as a concept slide, this is rather useful. So if every cell or if a cell was a sphere and you force it to become a pancake, then the surface to volume ratio must change. And to the extent to which the lipid bilayer of the surface of the plasma membrane is inelastic, the only way that the cell could accomplish such a shape change is by either having lots of ruffles, like a wrinkled cellophane bag that's covering the cell, and that simply unfolds, or by making new plasma membrane through lipid trafficking as the deforming stress occurs. And both mechanisms occur to varying degrees in different cell types. So that this lipid trafficking response is a key cytoprotective mechanism against the occurrence of plasma membrane stress failure. And experimental, and hopefully at some point, clinical manipulation of this exocytic response may help us precondition the lungs to become more resistant against deforming stress. That is sort of the goal of everything, of trying to understand these phenomena. This slide summarizes a number of papers by Nick Vlahakis and several years of work, and it's always humbling when you can take years of work and put them on one slide. But what we have learned is that deformation-induced vesicular lipid trafficking in alveolar epithelial cell is both a temperature and energy-dependent process. It requires intact microfilaments and microtubules. It involves both exocytic and endocytic pathways. It is substrate-specific. And if you do inhibit deformation-induced lipid trafficking, either by interventions that target the plasma membrane itself or the transport proteins and cytoskeletal proteins, then the likelihood of producing stress failure is much increased. And I think in the interest of time, I'm going to skip a few slides here to return to this. You've already heard the word caveolae. Caveolae are these invaginations of the plasma membrane, which are both signaling platforms. They are enriched in sphingolipids, cholesterol, but they're also a source of excess membrane for unfolding. These caveolae were initially described in endothelial cells, but it turns out that alveolar epithelial cells, particularly type 1 alveolar epithelial cells, are very rich in caveolae. In a study that's been presented in abstract form, you can see that the expression of caveolin, which is a marker of caveolae, is actually a deformation-sensitive event. When you culture type 2 secretory alveolar epithelial cells and stretch them and keep them stretched, that promotes a more rapid transdifferentiation to the type 1 phenotype and enhances the development of caveolae, and therefore may, through this mechanism, affect the deformation sensitivity and response of the cells. I'm going to conclude with something that has occupied my laboratory for the last year or two, and that is to build a new toy which hopefully gives us new insights into the biomechanics and the lipid trafficking of plasma membranes. And this toy is an optical trap. Laser light, like any light, imparts energy. And Newton would predict that this energy is sufficient to move particles. And the diagram here gives you a rough outline why the diffraction of light that is out of center of a particle results in a force which ultimately traps the particle in the center of the beam. And you can go ahead and move, in this case, a micron particle, and move it in medium, if you will. Well, you can coat this particle with a ligand of interest, And if the ligand is to a protein that's associated with the plasma membrane, but not the subcortical cytoskeleton, then you can lift a lipid tether off the cell. And that lipid tether has a tendency to want to recoil. So you can measure length tension curves of lipid tethers. The way you can measure the force that this tether exerts on the particle is because that force will tend to move the particle away from the center of the trap. So the displacement of the particle relative to the center of the trap becomes a measure of the force that the lipid tether has exerted on the particle. And the force is roughly in the order of 5 to 10 piconewtons. And I don't know whether this is going to work here. Here we go. Well, I can't make it work, so we had it working before. It's all right. We can do this on the next slide. Oh. Here we go. All right. So you have a particle here that's held by laser light. You move the cell in apposition of the particle, and you see a lipid tether is formed here. This is not too healthy a cell, but it's gotten the most beautiful image of a tether for the purpose of demonstration. So what happens during this is, of course, that that lipid has to get recruited into the tether, otherwise you couldn't pull it off. The force that's exerted here is largely dependent on the adhesive interactions between the plasma membrane and the subcortical cytoskeleton, and has to do with the ease with which lipids are recruited here, meaning unfolding of residual lipids, and lipid trafficking, which occurs at the same time. So in the interest of time, I will focus only on this part of the slide. A typical lipid tether length tension recording will give you a readout like this, where it takes roughly between 5 and 10 piconewton to form the tether. But notice, once you form the tether, no additional force is necessary to keep pulling. And if you do it slow enough and long enough, tethers of up to 50 microns can be generated depending on the cell and the cell type. But there comes a point, there seems to be a stop, and there's an increase in the rate of force relative to the displacement or the tether length. And the slope of this relationship depends on the rate at which cells traffic organelles to the plasma membrane, mean grow new plasma membrane, and the rate at which you elongate the tether. So this is a technique that might help us define the rate constants for lipid trafficking from intracellular organelles to the plasma membrane, and at the same time study interventions that affect this trafficking. Some of those measurements have been made in fibroblasts. And you see that the kind of tracing that you get on the first pull differs from the third pull, from the fifth pull, because there is memory in the system. Lipids had trafficked during the first pull. So you end up with a cell that has more plasma membrane than it had before you pulled. A mechanotransduction event, which incidentally must have had very little to do with focal adhesions. So when I come back and pull again, the amount of plasma membrane for unfolding has clearly increased. This slope here, as I said, ought to give us some information about the rate constants, but this hypothesis has not been tested to date. So what I tried to do then in this presentation is convince you that plasma membrane stress failure is an important mechanism in the pathogenesis of ventilator-induced lung injury. It is by no means the only mechanism, but it is one that is likely important during overdistension injury, and as I will show you tomorrow, possibly contributes to low-volume injury as well, although by somewhat different mechanisms. Most injured cells repair plasma membrane wounds. If we estimate that for the lung, for this kind of experiment, about 60% of the cells repair plasma membrane wounds. Failure to repair wounds result in cell necrosis, and successful repair of plasma membrane wounds is associated with a pro-inflammatory gene response, and I will come back to this in my presentation tomorrow. Wound repair occurs through the trafficking and fusion of vesicular organelles with the plasma membrane, and deformation-induced vesicular trafficking is a cytoprotective mechanism which serves to maintain plasma membrane tension at sublytical levels. And I have to thank a lot of people who are actually doing the work and allow me to present it to you. In particular, I want to emphasize Randy Strutz and Bruce Walters, who spent the last year and a half building the optical trap, which we're having a lot of fun with these days. A number of graduate and postgraduate students in the lab, and now Nick Vlahakis, who's done most of the lipid trafficking work, and William Guilford, who helped us build the trap. He's a biomedical engineer at UVA, and Tik Bagano, who taught me how to spell lipid. Thank you very much.